Hello everyone, welcome to Wonders of Biology, where I explain interesting biology topics. Today I'll be explaining RNA transcription and processing. So let's get started. So before we actually talk about RNA transcription and everything, let's go into a little bit of the basics. So here we have DNA and RNA. So our DNA and RNA. And these two are basically the main ways of storing genetic information in our cell. So they're the main holders of our genetic information, and that's where all of our genetic code comes from. So DNA is the mastermind. It's basically the master code, codes for everything in the cell, and from this we transcribe our RNA. And you can see automatically that there's a structural difference over here. DNA is double-stranded and RNA is single-stranded. It doesn't have that double helix shape. How DNA stores all of this genetic code is through a sequence of these things called nucleotides. And you can see all of the nucleotides over here that are in DNA. And each of these nucleotides, they kind of come in pairs. So each strand has one and it pairs with the other on the other strand. So over here you can see these two are pairing together, those two are pairing together. And how this works is thymine always pairs with adenine, and then you have cytosine pairing with guanine. That's how they pair in DNA. What you will see with RNA, on the other hand, is you will not have any thymine. Instead, you will have this thing called uracil. Uracil basically just takes the role of thymine, it just replaces it entirely in RNA. So you would have uracil pairing with adenine, and then guanine and cytosine pair together just like in DNA. So the reason we take our DNA and transcribe it to RNA is just because it's a lot more easier to transmit like small sections of your genetic code when you want to use them and transmit that message to the entire cell. How this message is kind of transcribed and everything, basically you have your DNA, which is your master code. Your master code codes for how to make certain proteins in your RNA. So you would transcribe a section that you want to express, a section of your DNA that you want to show right now, and you would transcribe that into your RNA. Once you have your RNA, you want to take this message that's in the RNA and actually make it into a protein. And it's finally these proteins that lead to your expression of your certain genes. So your phenotype. So phenotypes meaning like your blue or brown eyes or your brown hair or your skin color, all of those things. So that's basically how everything works in general when it comes to cells and genetic information. Now that we have talked about all of the background information, let's actually go into RNA transcription. Here we have our DNA, our master code of the cell, which has all of the genetic information that we need. We want to transcribe a section of this DNA so that we can send that message throughout the cell, translate any proteins we need to, and thus express the phenotype that we really want to express right now. So in RNA transcription, we have this guy called RNA polymerase. Now, RNA polymerase is essentially in charge of everything with this process. It does all of RNA transcription from start to finish. So RNA polymerase basically sees this part of the DNA code that's highlighted in pink here, and this we call the promoter region. Another name we like to call the promoter region is the Tata box, and that's just because the promoter region is the section of TATA. That's the genetic code that's over there. So RNA polymerase sees this Tata box and decides to go over there, and it gets onto the DNA, it attaches over there, and it starts unwinding the DNA in that section so that it could start transcribing that DNA and get its RNA transcript. We like to call this process initiation because we are starting RNA transcription. So thus we are initiating RNA transcription, and that's why we call it initiation. Then we go into the next step of the process. So we started over here at the promoter region, and RNA polymerase basically goes along the DNA, transcribing what it needs to. So how RNA polymerase transcribes and comes up with this RNA transcript is it basically matches these these nucleotides that are just floating around in the nucleus. So it takes those nucleotides and it basically arranges it in the pattern that matches with this bottom strand of DNA. And this bottom strand we call the template strand because it's the strand that RNA polymerase is using as a template to make its RNA transcript. So this other unused matching strand, we call it the coding strand. So it's because we call it the coding strand because it matches the RNA transcript exactly. Because if you remember, DNA pairs very similarly to how RNA pairs. The only difference between DNA and RNA is that in RNA you have uracil instead of thymine. So the coding strand is almost identical to the RNA transcript, except it's going to just have T's instead of U's. So what RNA polymerase is making its RNA transcript off of is going to be your template strand, the bottom one in this image. And it goes along this template strand and it matches up the base pairs of what is given in the template strand and makes this RNA transcript you can see here in green. And the direction it codes this is going to be 5' to 3'. So this is the direction of the RNA transcript itself. So when it's coding, it goes from 5' to 3', the resulting RNA transcript that it's making. So this we called elongation because we have already started the process in initiation. Now we are extending or elongating this RNA transcript that we just started. So thus we call this step of the process elongation. So once we finish making our RNA transcript, RNA polymerase will hit this region called the terminator. So it's like a termination site, and that basically tells RNA polymerase that you are done transcribing the RNA, you have transcribed everything you need to transcribe, so it is now time to end making that RNA transcript and release it. So that's what RNA polymerase does. It hits this termination site, it rewinds up the DNA, and it just leaves. It goes away. Let's pretend that's RNA polymerase over there. It leaves, and it releases our nice RNA transcript, which is what we wanted with this whole process. So this is basically how RNA transcription works as a whole. So now we have transcribed our DNA into our nice RNA transcript. And now, depending on whether you're a prokaryote or a eukaryote, there's different things you're going to do. So you're either done or you're not done. So if you're a prokaryote, for example, so these are cells like bacterial cells, then you are done. You are done with this process. You have your finalized mRNA transcript, and that can be translated into a protein. So if I was to draw kind of like a prokaryote structure, we have all of our DNA in the center of the cell. We have no organelles. There isn't a nucleus to hold our DNA. So, and also prokaryotes have really less DNA, and that's because all of the DNA they have codes for useful things. Eukaryotes, on the other hand, we have like some random useless information in our DNA transcript. So, over here, we have our ribosomes just floating around in the cytoplasm. And remember, ribosomes are those things that are translating our RNA into our proteins. So when we have all of the DNA floating around here, we make our mRNA transcript from that. And then we're already in the region where the ribosomes are. So the ribosomes directly just attach on and they start translating that RNA into a protein. So there's no time here to modify that RNA. It's already in wherever it needs to be. So we can't really change anything about it. So we're already done. That RNA transcript is finalized and our ribosomes are ready to translate it. Now, on the other hand, if you are a eukaryote, so kind of like our cells, animal cells, plant cells, and all of those things. So if you're a eukaryotic cell, then you're not done with the process yet. There are still some things you need to do. So let's say this is a eukaryotic cell. So your DNA in the eukaryotic cell has this nice little container called the nucleus. So we have an organelle that can hold our genetic information in the nucleus. And we have our ribosomes floating out here in your cytoplasm. So the nucleus kind of poses like a barrier between the ribosomes and the DNA. So we can make our mRNA transcript, and then we have all the time we want to modify this and then send it out to the cytoplasm where the ribosomes can then translate it into proteins. So in eukaryotes, you have this whole modification process, and it's not only because you have more time here because of the nucleus, but also because eukaryotes tend to have a lot of useless information in their DNA. They have like these spacers in between all the useful information, and we don't want RNA transcripts having all that useless information because we don't want our ribosomes translating that part because it doesn't really code for anything. So we go through our RNA modification processing. So here we have our initial, for eukaryotes, here we have our initial mRNA transcript that we just transcribed from DNA. And you can see there's different sections called introns and exons. So exons here in these nice bright colors are our useful information. So we want to keep this. And introns, on the other hand, they are not. They are useless information. We want to remove those out. So the process of removing these introns and then stitching together the exons that are remaining is called RNA splicing. So we call that process splicing, and it is done by spliceosomes. So it's very similar sounding, so it's easy to remember. So it's done by spliceosomes. So now that we have our spliceosomes, they just remove all of our introns and then they stick together our exons. So we have this nice little order over here. So before we are done, though, we need to do a few more things to our mRNA transcript. So we add this thing called a 5' cap, a 5' GTP cap. Honestly, it's fine if you remember it as 5' cap, but it's called a 5' GTP cap, so it's added to the 5' part of our mRNA. And then we have a poly A tail added to the 3' end. And it's called a poly A tail because it's a bunch of A's just after each other. It's like so many little A's after each other like that. So that's poly for multiple A tail. So it's a poly A tail on our 3' end. Our 5' end has a GTP cap, 3' end has a poly A tail. So why do we add this 5' cap and this poly A tail? What is the point of that? So what the 5' cap and the poly A tail does is it basically tells our nucleus that, okay, this mRNA is completely finished and it's ready to send out to the cytoplasm. So without that 5' cap and the 3' poly A tail, the RNA is not sent out to the cytoplasm because it can't pass through the nucleus, because the nucleus wants to make sure that all of the mRNA is ready before it sends it out to the cytoplasm like that. So over here you can kind of see that there are multiple ways we can do splicing. So since in eukaryotes, we like to multi-purpose our genetic code. So basically, these same exons can code for multiple proteins depending on which ones we use and decides to disclude. So we can maybe remove our third exon and then we can get this certain transcript, or we can maybe remove the first exon and get a different one. So based on which ones we decide to use, like let's say we use these 2 exons, it would be one. If we use these 2 exons, we would get another RNA transcript. And that allows us to code for multiple proteins with the same exact genetic code. And that way we can multi-purpose and use less space in our genetic code to make so many different proteins. So spliceosomes can also do that. So depending on what protein we want, we undergo alternate splicing and combine our exons in multiple patterns to get different proteins. And this is very useful, and it's only a eukaryotic thing. Prokaryotes don't have that modification, so they can't multipurpose their genetic code like that. So that's it for today, guys. Thank you so much for listening, and I really hope that helped. If you have any questions, you are welcome to put it in the comments below, and see you next time. Bye!